I see. Yep. And how many of you are familiar with differential equations? Now, you've had some differential equations already in the course as well, right? Okay, good. So I think we're going to get along just fine. What I'm going to do is I'm going to be switching back and forth between the slides and the software, and software running on another machine somewhere else, and the blackboards. And what I want you to do is to feel absolutely free to stop me at any moment and ask questions. I really want this to be interactive. So let's begin. So the name of our software is Plaza Sur. Plaza Sur is one of the very small islands in the Galapagos. And it's called Plaza Sur because we are treating the current version of our software as a pilot. And so this is the small version. The full version, once it's done, is called Daphne. And if you are familiar with The Beak of the Finch, have you read The Beak of the Finch? It's a wonderful book by Rosemary and Peter Grant, who had a 10-year project on the island of Daphne. They very carefully measured the beaks of finches over, or actually 10 or 20-year period, I think it's 20 years, and took careful notes about the vegetation that was available and witnessed evolution in process on Daphne. And so that's really what we are all about here. We want to understand the evolutionary process that drives affinity maturation. Our ultimate interest is in vaccinology. My group works with a number of other groups in immunology and vaccinology to develop new ways of designing vaccines. And so all of this is towards those ends. A lot of people are involved with this, and I want to give them credit first before I run out of time at the end and neglect to give them the credit they're due. This project started at Duke University, and then I moved to BU, and that's where it's now quartered, but we have a lot of people still working at Duke. So in my group, which is 85% computational, Grace Kepler, Feng, and Sanjeev Gupta have put a lot of effort into this project. At Duke, Cliburn Chan, Harold Schmiedel, Eric Monton, and Anna Maria Mashi. Our experimental colleagues are Garnet Kelso and Masayuki Karaoka. They're immunologists, and we'll talk a lot about their work here. Steve Wallace in biomedical engineering, Jason Hawk at NC State University, Ann Haberman, whose intravital microscopy we'll be taking a look at, and Phil Hanna at the University of Michigan. So the project is supported by the National Institute for Allergy, NIAID. Yeah, I know, I got halfway through and I can't remember what it stands for anymore. And we are going to have a finished product at the end of the 5-year period, and you're going to help us to develop it by giving us feedback at this very early stage. So, as I said, I want to talk about affinity maturation. And affinity maturation, I think, is perhaps the key process for humoral immunity, for eliciting protective vaccines. And you may remember back in 1964, this paper, Eisen and Siskind. Do you remember the excitement around that paper at the time? Neither do I. I was 6 years old. I was precocious, but not that precocious. Is there a pointer up here? No? You can use the mouse. Yeah, that's true. Well, actually, I'm close enough I can just go like that. The only trouble is when I go over to here, the light shines in my eyes, but it'll work. This is a paper by Eisen and Siskind. Eisen and Siskind were immunologists. At the time, they had just developed a new technique by which they could measure the affinity of antibody in serum. Okay? It was a new biochemical technique, and they wanted to apply it to vaccinated rabbits. And so they vaccinated rabbits, and then they measured the bulk affinity of rabbit serum against the antigen they immunized with. And here's what they found. I'm just pulling out a specific row here so you can focus your attention on it. So at 2 weeks post-immunization, they had an association constant of, this is actually 1.0. The decimal point didn't show up very well. Liters per micromole. And microliters per mole, going up to 6-fold the original value in 5 weeks, and 250-fold at the end of 8 weeks. Okay, so as you all know, it is this high affinity antibody that provides protection against re-challenge from the same organism. "20 years later, Martin Weigert and his colleagues were studying mouse kappa chains and noticed that if they isolated these chains here, they all seem to be a lot like V kappa 21. And in fact, they were able to reconstruct all of the sequences that they isolated by assuming that point mutations were being introduced into the germline version of that receptor, and deduced that somatic mutation was occurring. Mutation was occurring during the immune response, right? And they put 2 and 2 together and figured out that somatic mutation was driving affinity maturation. So how does that work? This is a paper from Garnet Kelso's group, and he's showing something very similar. This is the unmutated version of the genes that he's studying, and he finds these mutated versions. And he looks here and finds an increase in affinity in these very cells. So finally putting 2 and 2 together, showing directly the somatic mutation and the affinity maturation are indeed occurring at the same time. So, you know, what is going on? What's going on is that after immunization, B cells that are activated go to a place called the germinal center, or I should be a little bit more careful about that. They go to the secondary lymphoid tissues, whichever is appropriate for the site of immunization, the lymph nodes or the spleen, and they begin to interact with T cells, and they form germinal centers. And in the germinal centers, the genes acquire mutations, and then selection occurs. Okay? So the genes that change and encode by chance new antibodies that bind the antigen more avidly confer an advantage on the B cells that bear them. And those B cells either multiply faster or die more slowly and eventually come to dominate the population, and so the average affinity rises. And these B cells also go on to become memory cells and long-lived plasma cells. This is another example of affinity maturation from our own group, showing affinity maturation to Bacillus anthracis, protective antigen. We're looking at curves with alum used as an adjuvant and curves with the antigen only. You can see that the alum boosts the affinity maturation substantially, and control, which is all over the place. So this is a cartoon version of the germinal center reaction. We'll look at real pictures soon, but let's get the cartoons first. They're easier to understand. So this is the germinal center. And in the context of the lymph node, what we would find is the T cell zone down here and the B cell follicle surrounding the germinal center, and the capsule up here, the edge of the lymph node. And this is the so-called dark zone of the germinal center. And here's where the cells divide very rapidly. The cells there do not express a lot of antibody. And cells from the dark zone divide, and at some point, they exit the dark zone and they enter the light zone. And in the light zone, they encounter follicular dendritic cells. And the follicular dendritic cells are specially made to sequester antigen. They have Fc receptors and complement receptors on their surface, and they hold immune complexes. Immune complexes are antibody and antigen stuck together. And so these cells are decorated with immune complexes. B cells that now have re-expressed their antibody molecules, their B cell receptors, can interact with the follicular dendritic cells, acquire antigen from them, and then interact with pardon me, interact with cognate T cells and receive signals to survive, proliferate, to go on to become either plasma cells, memory cells, or to recirculate back to the dark zone. So that's the storybook version of events. What we're going to do is try to put some mathematical precision into this story. So let's talk about this a little bit. Selection. All of these evolutionary processes involve a combination of mutation and selection. The generation of diversity and then selection from among the diversity that is generated, right? And the mutation part is pretty straightforward. What we know is that the antibody genes experience a very high rate of mutation. Point mutations are stuck into the genes. But the part that's not quite as straightforward is selection, and that's what we're going to be really thinking about. We want to understand how selection occurs, and this is very much an open question in understanding the germinal center reaction. And something that I think, if we can understand better, will give us a much better handle on vaccine development. So, as I said, you can think through how this must work. B cells generate exactly one antibody molecule each. The antibody molecules themselves don't change, although back in the old days, that was one of the ideas. People thought they could. Now we've ruled that out. So it can't be that the antibodies themselves change. It's the frequency of B cells that changes. So they must either proliferate more rapidly or die less rapidly. And that's what we're going to be thinking about. So here are some real pictures. These are images from Garnet Kelso's lab. And let me try to point out what we're seeing here. This is 5-color immunofluorescence histology. And what we're looking at is B220 in blue here. So what does B220 mark? I know. I should have covered up the damn legend. I'm not very good at this. Not very good at this. All right. Yes, those are naive B cells. Well, let me put it the other way around. What do you think we're using to mark T cells? Those are all extremely good answers, but in fact, we're using TCR beta. Those, yeah, okay. How about, what do you think germinal center B cells are being marked with? Actually, yeah, these are all good answers, but we're using GL7. See, it doesn't work this way around either. Okay, so I have to rethink this. So it's GL7 is being used for germinal center cells. Plasma cells are shown here in cyan, CD138, and FD cells, follicular dendritic cells, FDCM2, I think. So anyway, but here's what I want you to see. Here's the T cell zone in the center, the B cell zone surrounding the T cell zone, and then discrete individual germinal centers. And the FDC are not very easy to see, but there are a few scattered throughout the B cell zone. Excuse me, the germinal center. Okay, and the plasma cell foci are outside the germinal centers. Okay. These represent serial sections through the spleen, and what you can see is you can sort of reconstruct this in a three-dimensional way and understand what the organization looks like. And I think I just have one more showing a very, very busy section of spleen, but it's the same basic thing. Now, in addition to having these static images that give us very sharp, detailed pictures of the organization of the cells in the germinal center, in the last 8 years or so, we've been able to use intravital 2-photon microscopy. Have you talked about that yet in the summer school? Are there plans to talk about that here? Well, so let me just mention very briefly what it is. 2-photon microscopy is another form of fluorescence microscopy. So you know how regular fluorescence microscopy works, right? You use a laser, and the laser light excites your fluorophore to an excited level. And when the fluorophore relaxes back down to its ground state, it emits a photon, which you can then see. Okay? The trouble with that kind of excitation is that you have to use a high-power laser to excite your fluorophore, and if you try to get too deep into the tissue or you image for too long a time, it dries out and burns up the tissue. So what people figured out is you could use multi-photon microscopy. And what you do in this case is you have very, very high density of photons, but low energy. And this relies on your fluorophore absorbing 2 or more photons simultaneously and kicking up to the energy level. So you kick up to the energy level associated with twice the photon energy. Okay? So that means you can have a much weaker laser but a higher, that is to say, a lower energy laser, lower frequency, but much a greater density of photons. And you actually pulse the laser. You have a very intense pulse for just nanoseconds, and then relax, and images are collected this way. Now, the advantage is that you can image for a long time. The movies that we're going to see here go up to an hour. Anne is now working on imaging for 2 hours. The real problem is that the sample doesn't stay still enough over the period of 2 hours. And when I say sample, what I mean is you've got an anesthetized mouse who's lying there with his or her side cut open, exposing the inguinal lymph nodes, and we're directly imaging the lymph nodes in the intact living animal. Okay? And so this is what we're going to see here. This is a single germinal center. This is the bottom of the germinal center. So this is towards the dark zone, towards the T cell zone, and this is the top of the germinal center. Our B cells here are green, okay, and red are stromal cells. So the way this is done is that this mouse glows red. All of the mouse's cells glow red. And so what Anne did was to deplete the mouse of all lymphocytes and then reconstitute the mouse with green B cells and T cells, but not labeled. And then we take these images after immunization. So let's take a look at, let me see, can I get this? So these are blood vessels. The thinner stuff are stromal cells and collagen. And the B cells are moving around. The top is primarily the light zone. And while you can see things that are definitely vasculature like this finer stuff in here, these are follicular dendritic cells. These are non-hematopoietic. And so the mouse kept these guys, and they glow red with the mouse's own endogenous glow. And again, these cells move around. You can watch these things crawl around on top of and through the follicular dendritic cell network. And so really this is what we're going to try to understand. The germinal center is an extremely dynamic place. The cells are moving around continually. And they have to go from one place to another. They have to go from the dark zone to the light zone. They have to encounter follicular dendritic cells, take antigen from them. They have to then find T cells, and of course the T cells are moving around too. We'd see that here if they had been stained. And then once they make all those connections, they're going to differentiate, and then they have to go to the right place for whatever they've differentiated into. And so what we're going to try to do is to build a system that'll help us to understand this combination of things. So I want to stop here for a moment, because we're done with the overview of the biology, and we're going to start talking about model building. So are there any questions about the biology at this point? Yes?
In the previous movie that you showed, what's the, I know it probably is a time lapse, right? But
oh yeah.
What is the real time?
That's about 30 minutes. It's about 30 minutes. Any other questions? Yes?
Does it matter how you vaccinate the animal, whether it's IV or
Oh, yes. Yes, this...
how they interact and who might be presenting or maybe a macrophage?
Yeah, so these immunizations are foot pad immunizations, so that they will, because she wants to visualize the draining lymph node. Now, in terms of what cells are doing the presentation, I mean, once the antigen gets into the lymph node, and it's very likely draining directly to the lymph node as well as being taken by DCs. But we haven't studied the details of that. What we have seen, though, are we have seen the differences between intraperitoneal and foot pad immunizations. Of course, the intraperitoneal means that the reactions go on in the spleen, and foot pad means they go on the lymph nodes. We've seen substantial differences in affinity maturation under those circumstances. Oh, it does. The size of the immunogen also makes a difference. An immunogen that's too large can't drain directly to the lymph node, and it has to be carried by another cell. Yeah. Okay, so let's talk about modeling. Now, I'd like to think about the agent-based modeling or individual-based modeling that we do as not being a completely different way of doing the modeling, but being along a continuum of modeling strategies. Okay? And so How many of you remember your first class in fluid dynamics? Yeah, I know. Catherine and I are both physicists, and although I was a physicist much longer ago than she was, and I forget almost everything. I've been an immunologist for 20 years now, but I still get motivation from things in physics. Now, this particular thing in physics, if you study fluid dynamics, there's basically two ways to write down the equations of fluid motion. Okay, one in which you think of, so think of a river. Okay, river's a pretty good example of fluid. You can set a cage down in the river and sink it to a spot on the ground, and then think about what passes through the cage. The other thing you can do is you can put a floating box in the river and let it float down the river with the flow of the river. And these are two different coordinate systems, or two different ways of representing what's going on. The Lagrange version is the one in which the cage moves along with the river. And in fact, what you're doing really is you are looking at the river from the point of view of the substance that makes up the river. Okay, you're looking at it from the point of view of the individual molecules, if you like. The Eulerian view is the one in which your frame of reference is fixed, is a volume that is fixed to the Earth. And what you're doing is you're looking what passes through this volume. Okay, so I'd like to now think about ordinary differential equations and the kind of modeling that you may have been talking about thus far. So I noticed that Catherine was showing some viral dynamic models, right? And so in that case, what you're doing is you're thinking about a population of virus particles, or a population of T cells, which is an inherently Eulerian way of looking at it. You've got a box, and you're counting up how many individuals there are in the box. But you're not treating them as individuals. You're treating them as a population. You're looking at a density. Now, we could, and we're going to, and Catherine's already shown you one way of doing it, change our point of view and think of the individual entities as being the objects of interest. So instead of saying, here is a volume located at x equals 3, y equals 4, z equals 7, what is going on in this volume? I can say, here is a T cell, and its coordinates are the following. And I can follow the T cell around. And I can do that with 2 T cells, or 3, or 10,000 T cells, okay? And the point that I'm trying to make here, and I may be belaboring it a little too far, is that the two ways of modeling are not all that different. They really are just two ways of representing the same phenomena. And I think it's true that any physically realistic agent model is going to be mixing both types of modeling in it. What we're going to be doing is we're going to be focusing on the cell as the entity of interest. And so our model is going to be constructed of B cells, T cells, and follicular dendritic cells, and each one of them is an individual component. Each one of them has an intrinsic internal dynamics and moves around in the space, interacting with each other and giving rise to the phenomena that we are interested in. The model is inherently modular. I think this is another key idea in agent-based modeling. The point is that I have a model of a B cell, and I drop it into the model of the lymph node, and it knows what to do. The model is built in such a way that all of the inputs for the B cell are contained in the environment. And similarly, inside the B cell, I've got different modules that control different kinds of behavior. And I can drop those modules in, and I can replace them with other modules. I can build up new cells from modules, and I can build up new large-scale models, new tissue models by putting in different cells and so forth. And the point is that they all find the appropriate inputs and outputs in their environment. And of course, we can also drop in different modules to represent different levels of resolution, and I'll try to illustrate that. So this is my cartoon version of modular systems models, okay? So this shape represents a system. And the system is defined by two things. The subsystems within it, and those are these funny shapes here, and relationships among the subsystems, okay? And the evolution of this larger system is driven by differential equations of this form, where the relationships among the subsystems X change according to some differential equation. And the key is that these parameters theta, this is a whole vector of parameters, these theta are characteristics of these subsystems. And these theta are going to be changing according to a different set of dynamical rules. And we can plug in different versions of these systems as appropriate. So here's the real system, okay? Now we get back to immunology, except that now it's immunology from the perspective of modeling. So let's see how we put together a model for affinity maturation. So let's focus on the B cell. That's where all the action is happening. So this is a model, a relatively simple model for B cell differentiation. A naive B cell becomes activated. The activated B cell then self-renews, it divides. Some of the activated B cells become short-lived plasmablasts, which secrete antibody and go on to die. This is an apoptotic cell. Some of the activated B cells then go on to become germinal center centroblasts. These centroblasts are the B cells that divide rapidly, and that's indicated by this green arrow here. These cells are in cycle. Centroblasts then differentiate to become centrocytes. These are the cells that zip around in the light zone. The centrocytes have a higher probability of dying through apoptosis. They can become rescued centrocytes. And the way they become rescued is they have to interact with FDC and T cells. So I've colored this arrow purple because it's special. Each one of these arrows represents some process: differentiation, division, death. And this purple arrow represents the most complicated process on the whole board. This is what drives selection. And then once the centrocyte is rescued, then either recycles back to becoming a centroblast and begins to divide again, or it becomes a long-lived plasmablast and it does secrete antibody, or it becomes a memory cell. And that's the whole thing. What we're going to do is we're going to pick up the story here at centroblasts. We're not going to worry about what goes on down here. We're going to pick up right here. Okay. Now we have to start thinking about the individual things that cells do. What are the behaviors they do, and how are we going to represent them? So let me pause here and ask for questions about the differentiation model. Is this okay with everyone? Okay. So now our model is a model that's based on forces and biochemical equations. So, because we're physicists from way back, we do try to keep things in terms of Newton's laws and so forth. And so, when we talk about cell motion, what we want to do is understand how cells generate the forces they generate, and what appropriate representations are. So here's so a cell is going to adhere to the collagen matrix and pull itself along. And from observations that many people, including Ann Haberman, have made, the motions of cells appears to be quite random for the most part. It appears that they move in a given direction, and they stop and they turn around, and there's no particular direction to it. And so we have to model this as a random process. Cells exert forces on each other. And so there are two ways they do that. One is that cells crash into each other, and they're not allowed to pass through each other. Okay? And so there's a repelling force, and the repelling force comes into play only when they touch each other, right? So they touch, and suddenly a force is generated, and the more they try to move towards each other, the more they are repelled from each other. And actually, this turns out to be really important in the dark zone, because otherwise, everything collapses down into a rapidly dividing neutron star. The other kind of force they exert on each other is attractive. And what happens once cells touch each other, they bind, they have, in the place where they touch, adhesion molecules bind to each other. And so cells, once they've touched, form a bond, and then when you try to pull them apart, there's a force that tries to pull them back together again. Okay? And this has to do with the adhesive bond here, and so we have to model that as well. So these are the forces that act between cells, and it turns out that these bonds are going to be really important for what we're about to do, because the B cells, when they come into contact with the follicular dendritic cells, have to sit there for a while and talk to them. And we'll talk a little bit more about how that is modeled. But for now, just recognize that these are the two forces that cells feel. And in addition to the forces, they also feel the influence of chemokine gradients. Familiar with the concept of chemokine gradients? So you know that cells have chemokine receptors, and by detecting the difference in ligation of the receptors in the front and the back, the cells determine which is the front and the back. And cells will direct their compass to point in the direction of the greatest density of chemokine and move in that direction. And so we put that all together, and as you all already guessed, we have these equations. Now, I'm going to explain what these equations are, but I will not quiz you at the end. So this is position, v is velocity. dx dt equals v. That's the definition of the velocity, okay? dv dt is minus gamma v. So the change in velocity has a component that points in the opposite direction of the velocity. So if I'm trying to move this direction, there's something that's pushing me back. What's that? That is drag. Okay, so this is a very viscous medium at this scale. And so cells trying to move through the medium experience a viscous drag. P is the direction of the polarization, is the polarization direction. And so phi times P is the force that the cell itself is generating. And it's pushing the cell in the direction of the polarization. And F are the external forces, the forces that are due to bumping into and sticking to other cells, as well as to whatever else happens to be in the environment. At the moment, there is nothing else in the environment. And finally, the polarization. The polarization also experiences what we can call a pseudo force that wants to restore it back to zero. Skip over to here. This is the chemokine gradient that wants to align the polarization in the direction of the chemokine gradient. And this is a special term. This is a white noise. And so this is just, this is a force that just continually kicks the polarization vector. So the polarization vector is pointing in some direction, and it's being constantly kicked. And so it's kind of doing like this. And so this is really the key to the way the cell moves. This term is constantly kicking the polarization vector around. This is constantly resisting change in the polarization vector. And this is trying to get the polarization vector to point in some specified direction, some preferred direction. And this just describes how we get this direction field from the chemokine gradient. Now, the only thing I want you to take out of this, well, no, there's two things. This is the density of chemokine receptor. And you see that the force, or the pseudo-force that the cell feels, is proportional to chemokine receptor density. This is important because the chemokine receptor density is itself going to be regulated. And it's proportional to the gradient of the chemokine field itself. And then there's a term here that represents saturation, right? So are you familiar with this idea? If your cell is in a chemokine field with the concentration so high that both the front and the back have its receptors completely saturated, it can't tell where the gradient is anymore. And so this mathematical expression is just simply realizes that. That's how this comes about. So, the direction field increases as the gradient increases, but it decreases as the concentration increases. Okay. So let me. Yes.
Is it something related to osmosis, something like because gradient density moves?
Well, it's actually different from osmosis. Osmosis is a passive process driven by differences in concentration. Following a chemokine gradient is very much an active process, and it requires sensing the gradient and sort of computing what direction to go, and then engaging your locomotory apparatus to push you in that direction. So it's an active, yeah.
There also you have osmotic potential which drives the system.
Yeah. Well, and in fact,
it crosses my mind.
sure. And the beauty of mathematics is you can actually use the same equations to model both of them, in spite of the fact that they have very different underlying mechanisms. Let's talk, before I go on to the next thing, let's talk about receptor regulation. That's the last thing I want to talk about. Then we're going to take a quick look at the software itself, and we'll look at an example. So we have receptor regulation, and R is the density of receptor. Let's call it chemokine receptor on the surface of the cell. And what are the processes that the chemokine receptor is involved with that will change its density? It's fairly straightforward here. Yeah, exactly. So there's production. So the cell is making new receptor, and it's transporting it to the surface. So we have to have a production term. So we have dr dt is, pi for production. I like to use Greek letters for my parameters so that I can tell the difference between parameters and state variables. So there's pi, and you notice that I'm going to ignore anything else that goes into this. I'm going to lump everything else into production here, but we all know that in order to produce a chemokine receptor, we have to transcribe the gene, we have to translate the transcripts, we have to post-process the protein, we have to transport it to the surface. We're going to lump all that together into one constant pi, and that's going to be fine. Now, later on, if we think that those processes are going to be important, or if we want to look at a much finer temporal resolution, we can plug in a more detailed model for receptor synthesis. But for our purposes, this is going to be fine. So what else happens to a receptor, do you suppose? It's recycled. And so what we're going to say is that there is a degradation recycling rate. And again, it doesn't matter exactly what happens to it. The point is that it is no longer available in the membrane. And so there's a constant lambda, and it's first order in R. Okay? You can't remove what's not there, and removal is on a constant per capita basis. Finally, I'm going to put one more thing in there, and this is going to turn out to be very important. Now, the receptor can be ligated, and we ought to put in terms that represent what happens when the receptor is ligated. So there's a forward rate, R times the ligand, and if the ligand ever comes off, now R star is our bound receptor, okay? So the free receptor is created, is removed, is ligated, and so once it's ligated, it's not free anymore, so this counts as minus, and should the ligand fall off, it's free again. So that's what we have here. And now there's one more thing I want to put in, and that is an equation for bound. So is it synthesized? Should there be a term for synthesis of bound receptor? No, because bound receptor is not synthesized. The unbound receptor is synthesized. So the only place this comes from is from here. So there's K plus R L minus K minus R star. And one more thing that I want to emphasize, the ligated receptor is going to be internalized more rapidly than the unligated receptor. Okay? So once the receptor is engaged, an active process of internalization begins. And so we have an expression that looks like this. So what happens is receptor is made, it's transported to the surface, it swims around on the surface for a while, and then after a certain amount of time, it's pulled back in, or it degrades and the unfolded protein is pulled back in. It binds to chemokine, and of course it transmits signals once it's bound to chemokine. That's not in here because it doesn't affect the density of receptor. And once it's ligated, active internalization begins and the receptor is downregulated. Okay, so let's see if I can make this work. I want to go to the model itself now. Oh, I just recognized that I'm not plugged in and I have about 5 minutes left. So let me just grab my adapter over here. Or no, maybe it's over here. Yeah. It's over there. That's going to be... Oh! And I guess I had a model up here, but I have to restart it. Okay. Okay, so now we're gonna take a quick look at the simulation system. And I'm not going to spend a lot of time describing how to work it just yet. What I want to do is just to illustrate what we've been talking about. This afternoon I can come back and talk about how to actually run the thing. For now, I'm just going to load a scenario that I've already created, T cells only. And so what I've got here is a collection of T cells. And these are, we can think of them as being follicular helper T cells because they are going to have CXCR5, which is the chemokine receptor for CXCL13, which is the B cell follicle chemokine. And there are 50 T cells there. And we're just going to, let me just see how long this is. Okay, this is a 2-hour simulation. Let me see what my options are. I actually want to write it and don't need to run that. Okay, so now the other thing is that I've got a field of CXCL13 in the center. So it's peaked at the center and diffuses towards the outside. I don't know if I can show you that or not. Let me see if I'm going to be able to see it. Well, this shows you where it is. You can't actually see it, but this shows you what the extent of the Gaussian is. Although it's not cube, it's actually spherical. And let's just run it and see what happens. So it's just like a swarm of gnats, and they're just moving around. Let's see what happens if we change the details of the way the T cells regulate their chemokine receptors. So CXCR5, and so what we're having, the degradation rate for the unbound receptors is a rate of 1/100 minutes. Internalization rate is 1/10 minutes. So let's suppose that the cells don't internalize their receptors at all. And we'll rerun this. And we'll call it T cells only 2. What's happening? They come into a clump. Why is that? Well, the reason for that is that there is a peak concentration in the center, and the T cells are climbing the gradient, and every one of them climbs the gradient, and they are packing as tightly as they can given the repulsive force that they've been given. And what is the effect of having receptor regulation by ligation? How can you explain the difference between what we've just seen and what we saw in the first one? Let me just show you that one again. Every time we run it, it's a different run. So what we do is we ask it to put the cells in at random, and then everything runs. And because it's so stochastic, we'll get a different result. But of course, it's going to be statistically similar. So there's a striking difference. The only thing we've changed is the receptor, the ligated receptor internalization rate. Yeah. Exactly. So by downregulating ligated receptor, the total amount of receptor that's available for sensing is lower, and the total response to the gradient is lower. And so the cells that are near the peak of the concentration don't feel it as much as the cells that are towards the outside. And so what actually happens here is that the cells that are nearest the center downregulate their receptor, and they begin to drift away because they're not feeling it anymore. By the time they reach the outer edges, their receptors are upregulated again, and they begin to feel it. And so the effect is that it's as if the cells are confined within a zone within which they're free to move around. So they're not continually forced to the middle. And in fact, this is what we see, and this is our hypothesized reason for that. Okay. One of the things that one of the exercises that we'll do this afternoon, for those of you who choose to play around with this, is we're going to see if we can get cells to oscillate back and forth between two regions. So if I have T cells, oh, by the way, I need to keep track of the time here. Okay. Suppose I've got CXCL13 here and CXCL12 here. And of course, I mean, this is just, they interpenetrate, right? These are fields that go into each other. And if I've got cells that express CXCR4, which matches up to CXCL12, and CXCR5, which is the receptor for CXCL13, and they both regulate their receptors in the way that I've just described. Think about this. You could have cells here in CXCL12, and CXCL12 downregulates CXCR4, but not CXCR5, and so they drift up here, and then the opposite takes place. And so the cells should move back and forth between the two zones. We'll see if we can get that to happen. Oh, thank you. Okay, now let's get on with the actual germinal center reaction and describe the rest of the system. Okay, so what else do we have to have? We have to have somatic mutation. And so in our system, every B cell has a heavy chain and a light chain, full DNA sequence, and every time a B cell divides, it introduces point mutations into the heavy chain and light chain. And some of you may know that the somatic mutation mechanism is mediated by an enzyme called AID, right? And AID has a sequence specificity. Certain DNA sequences are more prone to mutating than others, and all of this is put into the model as well. Furthermore, the mutation rate may vary throughout the course of the germinal center reaction. We have good theoretical reasons to believe that it does, but we also know that AID expression is upregulated by contact with T cells. And so we believe that it's likely that during the course of the germinal center reaction, when B cells interact with T cells, they have an upregulation of AID expression, so the mutation rate changes throughout. And these are parameters that we can change. Now here comes the part that is, oh, sorry, we already did that. Here's the part that is the most uncertain in the entire model, and it's because we do not have the information required to supply this part of the model. And this is where a lot of our guessing and a lot of our computational work outside of this model per se goes. So what do I mean? I've got a DNA sequence. It encodes an antibody, And suppose I couldn't, in fact I can. I can use SPR, I can measure the on rates and off rates for that antibody binding to its antigen. Now, I introduce mutations and I change 1 or 2 amino acids. What's that going to do to the affinity? What's it going to do to the on rates and off rates? Really hard to know. In fact, this is a really active area of research right now. And so if we're going to simulate the germinal center reaction, we have to put something in there for that. And so what we're going to do at this stage is put in a relatively simple model for how the affinity changes when you change amino acids. And here's what the model is going to look like. We're going to look at the logarithm of the affinity, or the association constant. And we're going to look at how this quantity changes with the single amino acid substitution. And so this is 0. This is no change. This is increased affinity. This is decreased affinity. And so here's what we're going to do. Whenever we see an amino acid change, we're going to pick a new affinity at random from the distribution I'm about to show you. So there is a probability of having an increased affinity, but there's a greater probability of having a decreased affinity. And so this probability declines exponentially, and so does this one, but this one declines faster. And the rate of this decline is called lambda plus, and the rate of this decline is called lambda minus. So if you change 2 amino acids, there's a formula that tells you how to get the appropriate distribution from this one. And so this is our very simple model. And one of the things that we can play with are these parameters. Make it more or less likely to get large changes. Now there are a lot of changes that occur that are categorical changes. Change not continuous. So what does change continuously? Well, the receptor density changes continuously. The concentration of antibody in the environment changes continuously. The position of the cell changes continuously. But some things don't change continuously, and that is the phenotype of the cell, right? So one moment you've got a centroblast, and then at some arbitrary time later, bang, it's now a centrocyte. Now we recognize that that's not how cell biology actually works, that there is a transition, but we, like immunologists do all the time, are going to use these as placeholders, as representations of a whole suite of changes that go on inside the cell. And so cell division is another example. At one moment you've got one cell, a moment later you've got 2. And so these changes are discontinuous, and so what we do is we have a probability that that discontinuous change occurs at any given time. And the way it's modeled is that every discontinuous change is treated as if there's a master control molecule that pushes that change, that makes that change happen. And so there is a molecule called SDiv for division signaling molecule, and there is an underlying biochemical set of rate equations that govern the behavior of SDiv, which is continuous, and it's going to interact with the other components as well. So there's going to be division, there's going to be several different differentiation signals, apoptosis signals, and so forth. And they will interact with each other, and there will be a set of differential equations that describes how they influence each other, and then each of these individually triggers a specific categorical change or discontinuous change in the cells themselves. And here's how that system is set up. It's it's really not that complicated. And so we have a molecule called activator, which we treat as if it is given to centroblasts at the time of their creation. And activator does three things. It inhibits differentiation of centroblasts to centrocytes. It drives cells through cell cycle and into division, and it inhibits apoptosis. Activator is not being actively synthesized by the cell. Activator is being preserved. So what happens to activator? When the cell divides, half of the activator goes to one daughter, half goes to the other. So as the cell divides, the quantity of activator is split in half every time, and eventually activator falls below the level at which it's capable of doing these things. But it's not a threshold. These are rate equations. And so when activator falls below a certain level, division stops. The inhibition of differentiation to centrocytes is released, and the inhibition of apoptosis is released. And so suddenly it can do either of these two things. Now the centrocyte, and this is just sort of a cartoon. We think of this as being mediated as an enzymatic reaction. So activator is an enzyme, and there is a div molecule that is activated by activator in an enzymatic reaction. All of these things are modeled as if they're this kind of enzymatic process. So we now have a centrocyte, and part of what happens when the cell becomes a centrocyte is it downregulates CXCR4, upregulates CXCR5, and the way that's accomplished is the synthesis rate is changed. And so it moves out into the light zone and interacts with follicular dendritic cells, which then allow it to ligate the B cell receptor. Ligated B cell receptor induces signal 1, S1. Okay? And S1 is being generated the whole time that the B cell and the FDC are in contact with each other. And once they come apart, S1 begins to decline. The activated form of S1 begins to become deactivated. And if it bumps into the T cell, while S1 is still sufficiently high, the T cell bears CD40 ligand. It binds to CD40 on the B cell, ligates it, and the CD40 ligand plus S1 forms signal 2. And signal 2 then drives differentiation and rescue. The centrocyte is now inhibits apoptosis again, and it is able to either go back to being a centroblast or go on to become a long-lived plasma cell or a memory cell. And if it doesn't bump into an FDC in time, or if it bumps into an FDC but doesn't have sufficiently high affinity to take antigen from it, it never gets BCR. It never gets BCR ligated, and so it never generates S1. Oh, and I forgot to mention it before, but once S1 is there, S1 also inhibits apoptosis. So it's a temporary savior from apoptosis. If it doesn't get that, he never gets S1, and apoptosis eventually kicks in. If it doesn't get CD40, then eventually S1 drops, and we get apoptosis again. So that's how all of this goes. And in the interest of time, I'm going to skip over a description of the FDC B cell synapse. And well, I may come back to that depending on what we get. We've already talked about adhesion. I'll come back and I'll talk about the synapses, but let's look at the output of the model first. And so this is what the model output looks like. This is over the course of 20 days, And I want to tell you immediately that this is not what an actual germinal center is going to look like. So we're still playing with the calibrations, and I've got some others that are a little bit closer. But so this in black is the number of centroblasts. In blue, the number of centrocytes. And in red and green, the number of long-lived plasmacytes and memory cells, respectively. And so what we see here is that we have a burst of growth, and then activator begins to become dilute, and growth drops. But then at this point, what happens is that centrocytes are being rescued, and some of them are being driven back into the centroblast state, and so we have another round of cell division here, and they're clearly distinguished in these pictures. And you can see that you can even see here's centroblasts, centrocytes, and then these guys are directly related to the centrocytes here. The whole thing's about affinity maturation. So do we have affinity maturation? The answer is yes, we do. This is 10 to the 6th. This is 1 micromolar, or roughly 1 micromolar. It's actually it's 1 molecule per cubic micron, but it's essentially a micromolar. And by the end of the process, it has increased by a factor of 10. So there's a 10-fold increase in the affinity, and for the NP system, that's just about right. Another quantity of interest is how many distinct antibodies have we generated during this time? And what we see is that around day 5, we begin to see variant amino acids. Of course, there's going to be many more variations of the nucleotide sequence, but those are not important. And by the time we're done, we have about 150 different antibody molecules. This is another variation showing what is a more realistic kinetics of the cells. But when we have a more realistic kinetics of the cells, we get a less realistic affinity maturation. You see, so we're still trying to figure out exactly where the changes need to occur. And what's important to realize here is that the mechanism of selection is not known. So this is not something we can just, go to the literature and figure out. I can't see it at all, Jean. Thank you. So what we have to do is make hypotheses and put the hypotheses into the model and see what we get out, and see whether the parameters that we have to use to get a hypothesis to work make sense or not. And so that's where we are now. This is in a period of calibration. What I want to do finally is to show you one of these models running. We'll see if it works. I've got so these are relatively complex large models, and so they take a few hours to run. And I've got a couple of these running on a server back in Boston, and that's something that you'll want to. Okay. Let's see, can we move this over here? No. Oh, I know. I guess I can't. I can't show you the other server. It's too bad. Nope. Well, here, come on up here and gather around. No, that's not a good idea. We'll figure it out for this afternoon. But as you saw at the beginning, we had a little trouble figuring out how to get what's showing here to show up there. But I can tell you, it's amazing. So I have 3 more minutes. Let me see if I can. Oh, here, try this. There we go. There we go. Okay, good. Now let's see. So this one is one that's still running. And you can't see all too well, but red are the B cells. This is the dark zone. Green are the T cells. And what we're doing is we're taking a snapshot every 3 minutes. And so a cell actually moves a fair distance in 3 minutes. And so it's hard to follow individual cells. And the orange guys here are follicular dendritic cells. And we're just marking the center of the FDC with the ball. The actual follicular dendritic cell has an extent that goes out about 25 microns. The balls themselves are about 5 microns. And their sphere of influence goes out like that. Let me see if I can show you one other, which is already completed. Yeah, okay. And I think this one no, okay. Oh yeah, here we go. Well, I think what we'll do is we'll just wait till the afternoon when I've got the computers hooked up more reliably and show you how all this works. I'm just going to abort this and see what happens. Now, okay. Yeah, so that's what we'll do. So we'll look again this afternoon at the system. Now, I want to give you a warning. The system that has been installed on the machines here is a smaller version of what I'm showing you, because the machines in the lab are 32-bit machines, and the system was designed as a 64-bit system. And so to make it a 32-bit system, there are some things that are not going to work, or are going to work very, very slowly. And so we can run some projects that you can think of on our servers in Boston. You can run smaller projects on the system in the lab. And if you have your own laptops and happen to have 64-bit laptops, you can download the software yourself and run it directly that way. Enjoy your lunch.